All right, guys, welcome back. We are continuing in Chapter 3 right now. What we've been doing up to this point is looking at the subcellular structures within prokaryotes. This is all Chapter 3 stuff. And also in Chapter 3, I'd like us to be able to differentiate not only between, let's say, gram negatives and gram positives, but also between prokaryotes as a group and eukaryotes, which are, of course, the kinds of cells that make us up. We're constantly, throughout this course, going to be trying to differentiate whether it's between eukaryotes and prokaryotes or whether it's between different types of prokaryotes. All right? This has clinical relevance. If you're in the healthcare field, you want to use the right antibiotic against the right prokaryotic organism. We don't want the drugs to adversely affect eukaryotes because, of course, that's what we're made of. So here is a pretty good list of some of the differences between eukaryotes and prokaryotes. Of course, I think it was mentioned in an earlier lecture clip that eukaryotes means true nucleus. So that's one of the many membranous compartments you're going to find in a eukaryote, that true nucleus, and then a bunch of other membranous compartments you wouldn't find in the prokaryotes that don't have a true nucleus. Another thing we noticed, we were looking at the fact that there is a nucleoid, which is really one circular chromosome. It's not within a membrane, versus a paired set of linear chromosomes that you'd find inside of a nucleus with eukaryotes. Now, not all eukaryotes are going to have the same number or size of these linear chromosomes, but they are typically linear. We have a different packaging and winding of these genetic material. There are no proteins called histones around which genetic material is wrapped when you look at a prokaryote, versus in a eukaryote there are. All right, I mentioned the fact there are no organelles, no membranous compartments, in other words, in prokaryotes, but you do have them in eukaryotes. Another thing we've seen is the fact that there is a cell wall made of peptidoglycan. We've gone further with this because we know now that there are some prokaryotes that have thicker walls than others, and a couple others like those that cause walking pneumonia that may not have any at all. But when you find cell walls, they're made out of this peptidoglycan, a protein and a sugar. Versus in a eukaryote, well, some eukaryotic cells, like plant cells, have a cell wall. So it's not like they're lacking cell wall necessarily, but it's certainly made out of different material. It's more of a polysaccharide cell wall made of cellulose, not so much protein, all right? And of course, animal cells have no wall at all. And finally, we're going to see a difference between these eukaryotes and prokaryotes in the way they replicate themselves. Binary fission is the method of division when it comes to prokaryotes. We're going to really give that a lot more attention when we get into population growth, and especially when we get into the genetics of it, because we're going to have to get into some microbial genetics. Versus mitosis is the way that a eukaryotic cell replicates itself. Okay? So this is a pretty good list, and what I want you to check out is maybe in Chapter 3, Table 3.2, in your online textbook there, they have a lot of details, some of which I just mentioned with you guys. We have the binary fission versus the mitosis, big thing that we see as a difference within the cell division category. I mentioned the singular circular chromosomes that we see in the bacteria and archaea, which are prokaryotes, versus multiple linear chromosomes when you look at the eukaryotes. So definitely you'd want to know those things that I've talked about. There are some things maybe on this list that are not quite exactly what I talked about, but a lot of these things I think I have mentioned, all right? So make sure that you check it out and read a little bit about it in that section. These are part of your readings and fair game for some of these questions on the exam. Now, one thing I will mention, though, just because they have a bit more detail than I'd expect you to know, is you'd want to know that the membranes of prokaryotes are different than those in eukaryotes. And beyond that, I'm not so concerned that you know specifically what those differences are. There would be different kinds of phospholipids and different kinds of proteins, and that's about it. I'm not really worried about you knowing is there ester-linked bonds or ether-linked bonds, these different chemistry kinds of linkages. Not so important to me, but knowing that there are differences in the phospholipids and proteins is probably a good thing to know. All right. So here's a eukaryotic cell. We've got that nucleus in the middle there, and it's containing our chromosomes. We have these membranous compartments, the endoplasmic reticulum. I think a lot of these you guys have heard of because you've had A&P or you've had some kind of basic bio class. These are membranous compartments that get the work done within the cell, kind of like organs within the human body, right? These are organelles. They're going to have ribosomes because they need to generate proteins just like any cell would need to have. But these are larger ribosomes. These factories are 80S ribosomes. They're still doing the same functions as the 70S ribosomes that prokaryotes have. They're still producing proteins that will perform certain functions in the cell. We have lysosomes in here for digesting things. But there are two organelles I want to focus on a little bit more and give a little more energy and attention to. And those are the mitochondrion and chloroplasts. Chloroplasts are found in plant cells and they're not found in animal cells, but mitochondria are found in both. So here we have the mitochondria that have this kind of a yellow color here. This is the location for cellular respiration. A good portion of cellular respiration takes place here. And of course, back in grade school, probably the mighty mitochondria probably was kind of the phrase people might use. It's the engine for the cell. And you're going to also see chloroplasts in plant cells. All right? And this is where photosynthesis occurs. Kind of the opposite process. With cellular respiration, there's oxygen being consumed and energy is being liberated. In the case of the chloroplast, the oxygen is actually going to be incorporated into sugar molecules. So we're actually kind of going the opposite direction, taking CO2 and oxygen and making sugars out of them. So we have two diametrically opposed processes going on. These are absolutely structures you would find in eukaryotes. Now, one of the things that the reasons I want to pay a little more attention to the mitochondria and the chloroplasts is because when we zoom in and look at these particular organelles, they're kind of special in comparison to other membranous compartments. Other organelles that have membranes often have one membrane, with the exception of the nucleus. A lot of them have one membrane that contain whatever's inside of them. But when we look at the mitochondria here on the left, or the chloroplast here on the right, we actually see double membranes. We see, like here with the mitochondria, we have a membrane on the outside, and then I'll just trace it in red here. So what I'm tracing in red is a membrane that is an inner membrane for this mitochondrion, okay? And we also see similar situation with chloroplasts. Now, when we look inside of these cells, we also see that there are single circular chromosomes. Now, this sounds familiar, right? We see single circular chromosomes within prokaryotes, and yet these are subcellular compartments in a eukaryote, right? But they have their own DNA, basically. Okay, they have their own genomes, and they have 70 S ribosomes inside of these organelles. Okay? They have actual protein factories that are the same size as we see in prokaryotes inside of these subcellular compartments. And we find that these subcellular compartments can replicate themselves within the cell. Okay? So you'll see more of these mitochondria where you, like in the human body, where you'd expect there to be a need for more energy. So muscle cells and cells like that are more energetic would need more mitochondria, and in fact, you would see that. All right. Now, another thing that I don't have listed here, but is absolutely true, if you were to zoom in on this red inner membrane and consider the composition of this membrane, you'd find phospholipids and proteins like you would in any membrane, but we would see distinct similarities between the proteins found on this inner membrane and the proteins found on prokaryotic membranes of certain types. All right? And where do these proteins come from? Well, they're coded by the genes, right? Well, it so happens that when we look more closely at the genetics of the single circular chromosome inside of these subcellular compartments, we find similarities in the nucleotide sequence between these subcellular eukaryotic structures and prokaryotes that exist today. Okay? Now, there are some big differences, but those sequences still give us some evidence. All right? And what this evidence leads scientists to believe is that these subcellular compartments were once free-living organisms, and that this larger cell that became eukaryotic consumed these smaller prokaryotic cells. All right? And they made a deal because these particular compartments actually really perform really important functions, like I mentioned earlier. The energy of the cell and making sugar for a cell if it's a plant cell. All right? So here we have what's known as the endosymbiotic theory. All right? So endo, inside, or something like that, within, right? Symbiosis, this is a kind of a relationship. It may be a beneficial relationship in both ways, right? And it's a theory, which means basically that we found so much evidence for this idea that it's practically a law in science. In biology, this idea that there's a symbiotic relationship, it's really solid, all right? In other words, we haven't really found any good evidence against it, okay? So it's likely that an early eukaryote, okay? So here we have a proto-eukaryote, okay? And most likely of a form that is similar to archaea, okay? When we look at the membranes of eukaryotic cells, it looks to be more similar to the membranes of archaea membranes. Okay? So here we have this early proto-eukaryote, most likely an archaic type of organism, and it came along and consumed in these events, kind of endocytosed basically, right? Grabbed on and pulled in these subcellular compartments, all right? So here we have a subcellular compartment that is from an aerobic bacterium, a bacterium that uses oxygen to get energy, all right? And we think that's where mitochondria came from. When it comes from a green photosynthetic bacterium, well, that sounds like what a chloroplast does for a eukaryotic cell. That would be an explanation for the chloroplast, all right? So this endosymbiotic theory basically is the idea that an early archaic kind of cell consumed bacterial cells of a different variety within prokaryotes, right? So one prokaryote prokaryote from archaea group is consuming other prokaryotes in the bacteria group. And this leads to this eukaryotic cell. Now, I mentioned really early in the introduction, this woman by the name of Lynn Margulis. And Lynn Margulis identified these similarities, and that's what she's known for identifying this. So this really kind of blurs the line of the evolutionary tree, you notice, right? This is basically saying we are not just did these archaic kinds of bacteria or archaic cells give an environment where we could thrive, but they also are part of ourselves, okay? Now, this is sometimes hard for people to swallow. I mean, even the idea of evolution is hard for people to swallow sometimes, especially if if someone is quite religious. And this is frankly hard to swallow for anyone, isn't it? Because we didn't see this happen. We didn't watch this occur, right? The idea is that this happened billions of years ago. But the evidence is very strong. Now, just to kind of think about this in a different way. If we look around today, can we find examples that are like this? And I would give you an example of this. There are cells within our immune system called phagocytes. So here I'm going to draw a big old phagocyte, right? And we're going to draw a bacterium. Okay, so here's a bacterium in purple, and this is a nasty bacterium that causes tuberculosis, let's say. You can pick any disease process you want. Well, it's pretty routine for these phagocytes to come along, identify that these cells need to be disposed of, and then actually wrap the membrane, their own very membrane, around those bacteria. What do we end up with? Well, we end up with something like this, where this phagocyte, right, has consumed this bacterium, and inside we have the bacterial cell. Now, does this happen in our body? All day long. All right, this is what the immune system is good for. That's what it specializes in for certain cells. And it's also true that bacteria, certain types of bacteria, can thrive in that environment. Even though you would think, well, this bacterium is going to be a goner, it's going to be digested and it's toast, right? Well, bacteria are pretty wily critters, and they do have mechanisms that allow them to evade that digestion process. And we end up with an outer membrane around an inner membrane. It just so happens that in this eukaryotic symbiotic theory idea, the benefit for both the cell inside, which now has food, has shelter, right? And the cell, the bigger cell, which now gets energy or sugars from a chloroplast, that this is such a beneficial environment that these cells thrive together, all right? So I think that's where we'll leave it for now, and the last thing we have to do is to check out some viruses a little bit, get away from the prokaryotes, and just look at some of the function and structure of viruses, all right?